Okay, we're taking a look at part 3 of cellular respiration. I'm going to wrap up a few things here in our third part here. Starting with metabolic integration, where we left off in part 2, you're going to take a quick look at how different types of nutrients get metabolized other than carbohydrates, such as glucose. Now, there's a lot to this. We're going to look at some of just the basics. We're going to take a look at how proteins can be metabolized, and a little bit about how fats can be metabolized. And ultimately, as a quick overview here, what you're going to see is that these molecules follow the same pathway: glycolysis, the pyruvate oxidation, citric acid cycle, and then finally the electron transport chain, titled oxidative phosphorylation here. They follow the same pathway, but they don't follow it from step 1 to the end like you see with glucose. So glucose follows the entire pathway, starting with the very beginning all the way to the end. What you see with things like proteins and fats is that they enter in the form of different intermediates, in different intermediate steps in the pathway. And what's important to recognize here is that what we really have is an ongoing series of reactions that can start at the very beginning or can start somewhere in the middle and then pick up from where they start in the middle and carry on to the end. So we don't have to start in the very first step. We can kind of cut in line, so to speak, and start in the intermediate series of reactions and carry on to the end. And that's essentially what we're going to see with protein and fat. They kind of skip further down into the line and carry on from there. And that's really the fundamental difference between these. So taking a look at how the breakdown of proteins could work, the catabolism of proteins. First of all, proteins, as you might recall, consist of a series of amino acids. So proteins are a great big long chain of amino acids. Here's a quick figure of a protein with a series of amino acids all connected to each other by these bonds called peptide bonds, a type of covalent bond. So we have a great big long string of amino acids. Each one of these circles represents an amino acid, and a protein consists of a long chain of these. Now, during the initial breakdown of your protein in the body, during digestion, those amino acids are broken down one by one to form the individual amino acids that we see here. This is a different structure, obviously, but this is representing an individual amino acid, to clarify. All amino acids have in common 2 functional groups attached to a central carbon here. So we have a carbon, which is actually bonded to a hydrogen, and then 2 functional groups here. Now, what we're trying to get to here is that these functional groups are going to be systematically removed. At least one of these is going to be systematically removed during protein breakdown. So one of these here is called an amino group, and it actually consists of a nitrogen and 3 hydrogens bonded together like this here. And one of the first things that's going to happen once the amino acids are removed from the chain is that amino group, sometimes called an amine group, is essentially going to be removed here. And what's going to happen is that's going to produce a ammonia molecule. Ammonia molecule is going to be the byproduct from the removal of this amine group here. What that's going to leave then is a carbon compound consisting of only carbon, hydrogen, and oxygen, in most cases anyway, which can be further broken down via a variety of different pathways. So essentially, you've got a lot of different amino acids, and one of the first things that's going to happen is you're going to remove the amino group from all of them. What that's going to do is lead to the production of a byproduct here called ammonia. Ammonia is toxic, so it needs to be neutralized, and the body's going to do that by converting it to a compound called urea, which is then later excreted in the urine. Okay, and this is a process is called a deamination. So that's one of the first steps in the breakdown of protein. And although there's a lot of variation with all 20 amino acids, this is going to happen with all of them. Okay, so what you're left then is with a basic carbon compound, like you might see here in this example, where the amino acid glutamate is broken down to form alpha ketoglutarate, which is an intermediate in the Krebs cycle. Okay, so in this example of glutamate, we see that glutamate has the amino acid or the amino group, removed, it's now converted to a carbon compound, alpha-ketoglutarate, and alpha-ketoglutarate will enter into the Krebs cycle as one of the key intermediates. Let's back up here just real quick so you can see that alpha-ketoglutarate occurs here midway through our Krebs cycle, or the citric acid cycle. Sorry for changing the terminology on you there. It's the citric acid, i.e. the Krebs cycle. So here's alpha-ketoglutarate. So although we show it as a kind of a continual process, the truth is alpha-ketoglutarate can come in from an external source, a protein, amino acid, enter here, and be utilized to then generate NADH here, ATP here, FADH here, another NADH here, and then be used to form citrate and convert it over and over again. So these products can enter in different forms from different sources and contribute to the products downstream, so to speak, from where they enter. And so when we look at how a protein works, the amino acid glutamate can serve that role as an intermediate here. Now, other types of amino acids can enter in different places. For example, the amino acids cysteine and glycine, similar but different amino acids, rather than being converted into alpha-ketoglutarate, for example, they can be converted into pyruvate instead. And there's a series of reactions here with multiple enzymes that will ultimately take an enzyme like cysteine here, convert that into a series of reactions to pyruvate. And now in the form of pyruvate, this can now enter into pyruvate oxidation and serve as the starting point for pyruvate oxidation in the Krebs cycle or the citric acid cycle. Okay, so once they get into this form, they enter in that process as an intermediate rather than the starting compound, glucose. Backing up here to the starting figure real quick, just to follow the logic here, we see a protein is first broken down to an amino acid. Amino acid has its amino group removed. The amino group removed, then the amino acid, depending on the type, can be converted to pyruvate, as we see here, or it can be converted into an intermediate of the Krebs cycle, such as alpha-ketoglutarate here. Another reaction that I don't actually get into here is that it can also be converted into acetyl coenzyme A, which can then be the starting point for the citric acid cycle itself. Okay, so there's actually 3 different fates that an amino acid can take depending on which type we're talking about here. This figure here shows you all the different amino acids and all the different intermediates that they can take on. And if you want to pause it here and take a look at that, you can. I'm not going to require that you memorize all of these, but this does give you a more complete overview of what you're looking at here. So there's 20 amino acids, they're all listed here, and you can see that they're all showing with arrows indicating where they would actually potentially enter into the cycle and what intermediates they can produce. So your body is complex, no surprise, and one of the complex things about it is protein metabolism. Proteins themselves are complicated with over 20 amino acids, and each one is metabolized a little bit differently. So luckily for you, I'm not going to make you memorize all those other than the first couple examples that we talked about there. Next, let's talk about the catabolism, the breakdown of fatty acids or fats. Fats, specifically triglycerides, consist of really 2 components. A glycerol, often called a glycerol head, which is what we see here in this figure, or what we see highlighted here, consists of 3 carbons. And then the fatty acid tails, which in a triglyceride consist of 1, 2, 3 fatty acid tails, which are a long series of carbon and hydrogen, primarily carbon and hydrogen atoms bonded together. Now these represent lots of potential energy, and the way they're broken down is done in really at least 2 kind of primary stages here. So in the first one, we have the breakdown of the glycerol head. Okay, so I'm going to go back and forth just a little bit. So first of all, the head is separated from the 3 tails. Okay, and essentially all these parts are going to be chopped up separately. So the head is cleaved off, and then you're going to have 3 separate tails that are all going to be metabolized independently. Okay, so that's the first part. When the glycerol is, when the head is removed, the glycerol head is removed, it can be converted through a series of reactions into an intermediate in glycolysis, which is referred to here as DHAP, dihydroxyacetone phosphate, DHAP for short. DHAP, if you go back and look at this section on glycolysis, represents the stage in which our 6-carbon glucose is split into 2 3-carbon compounds, one of them being DHAP, the other one being G3P, glyceraldehyde 3-phosphate. I know these terms are confusing, but the point is that we have an intermediate here right at the point where our carbon compound splits in glycolysis. Okay, and one of the things that happens here is our product splits glucose, which is converted to fructose 1,6-bisphosphate at this point. It splits in half. We get one of those products being glyceraldehyde 3-phosphate, the other one being dihydroxyacetone phosphate, DHAP. DHAP is then immediately converted into G3P, and then the pathway proceeds with 2 molecules of G3P. So you may have to revisit glycolysis on that. Point here is that our glycerol head enters right here in the form of DHAP. It is then converted into glyceraldehyde 3-phosphate and continues along the way. Okay, so a fatty acid head can enter in the intermediate stage of glycolysis where we are splitting. And that's a good place to enter, by the way, just to point out a quick little side note here. It's a good place to enter because we've bypassed the energy input. If you recall early on, glucose, it requires an input of energy. DHAP enters right after that, so there's no energy required at that point. And even better, it enters slightly before energy is actually picked up. So DHAP will actually yield us, 1 DHAP will yield us 2 ATP before it's converted further into acetyl coenzyme A. But it actually bypasses the energy input stage. So it's a more efficient form of energy than glucose. Okay, so following the removal of the glycerol head, what happens next is a series of reactions referred to as beta oxidation. And this is what takes place with the fatty acid tails. Backing up real quickly here, so we got the head shown here and here, and then the tails shown here, which consists of a long series of carbon and hydrogens. And what's going to happen is all of those tails are going to be chopped up independently into 2 carbon molecules referred to as acetyl coenzyme A, what we talked about earlier, acetyl coenzyme A. So rather than generating acetyl coenzyme A from each pyruvate, as we would do from glycolysis, we're going to generate acetyl coenzyme A from these fatty acid tails. Okay, and there's a different series of reactions to do that, but essentially each tail is chopped up, and the fatty acids are generated into acetyl coenzyme A. Backing up one more time, looking at our starting figure, we can see that the fats are converted into glycerol and then the fatty acid tails. Glycerol is ultimately converted. Here they're kind of taking a shortcut and they're showing it as G3P, but we know that that's actually dihydroxyacetone phosphate, which is then converted into glyceraldehyde 3-phosphate. So the glycerol ultimately is converted here into glyceraldehyde 3-phosphate, and then the fatty acid tails are converted into acetyl coenzyme A. Each one of those will enter into the citric acid cycle and be converted into NAD, ATP, and FADH2. Okay, so the idea then is that we generate energy in different intermediates. Now, to calculate those intermediate, the energy from those intermediates, you'd have to spend a little more time. We're not going to do that here. Some classes do that. You get into calculating the amount of ATP per fatty acid, for example, or per triglyceride. We're not going to do that here, but that is something that you could do. And one of the things, just to give you an idea, one of the things that you would find if you were to calculate energy totals is that typically fats serve as a more efficient form of energy. Fats can actually be quite more energy rich than, say, glucose can, and so the energy yield per fat is much higher than what you'd find in a carbohydrate. Proteins vary, but depending on the amino acid and the composition of the amino acids themselves. Okay, next we're going to take a quick look at the regulation of aerobic respiration. So one of the things that cells need to do in order to ultimately survive in the long run, talking about kind of the big game here of long-term survival as a species, one of the things that cells need to do is conserve energy. And one of the things that's important to recognize is that metabolic pathways are somewhat costly for cells to operate. In order to run these pathways, resources have to be consumed. So they consume organic resources like glucose, proteins, fats, and in theory these are somewhat limited. Okay, now we're kind of looking at the more natural form of life on the out in the wilderness, for example, or in an area that maybe doesn't have endless amounts of food, unlike, say, the American lifestyle. But presume here that we're in an area with somewhat of a limited resource, which has changed in the modern world, although we are still somewhat limited. It's not quite the same. But the idea is that our cells evolved under conditions with scarce resources, sometimes very scarce, and the ability to survive, presumably throughout the course of evolution, came down to highly efficient forms of energy conservation. And without being able to conserve your energy, you would lose energy, and that loss of energy could be the difference between life and death. And that's at least part of the theory of evolution, is that cells evolved mechanisms to maintain energy in order to survive longer. So anyway, the point is that we're burning resources. We need to conserve those resources and make them go as far as possible. Okay? So we don't want to burn resources without doing so for good reason. Okay, now burning these resources is obviously a benefit because we produce ATP, which is needed for the cell for a variety of reasons. So it's obviously good to do that so long as the ATP is needed. Okay, one of the things that can happen though is an organism gets a bunch of energy, a bunch of organic resources temporarily, produces the energy it needs temporarily, and needs to stop producing energy for that point of time because it has enough to actually carry out the cell the cell procedures, or the cell activity, excuse me, that it's needing at that particular moment. One of the things that can happen is when a cell already has ATP, burning more glucose, for example, will simply result in a waste of ATP. So the cell can only use so much ATP at one time, and continuing to burn more and more in the short term is actually a waste. It's kind of like if you're burning wood to heat your house. Imagine you have a wood burning stove, and let's say you've burned enough wood to comfortably heat your house up to a cozy 75 degrees Fahrenheit. You don't necessarily want to keep throwing excess wood on the fire, because A, you can get it too hot, but B, you're wasting the resources at that point in time. Your house is already nice and cozy. At least temporarily, you don't need to burn any more wood at that particular moment. Now, eventually the fire is going to die down, and you have to throw some more on there, but you're going to want to do that in a timely manner. You're going to want to kind of pace your wood burning in order to match the temperature rise and fall in your house. Okay, so that's kind of obvious when you use the idea of temperature and wood burning, but the same thing is happening in a cell. ATP levels are rising and falling, and cells are going to need to burn glucose like you would burn wood in order to kind of match the rise and fall of ATP. So burn glucose when ATP is low, and conserve glucose when ATP is high. And done correctly, the resource, the organic molecules, can be used efficiently so that they can get the most bang for the buck, so to speak. And so the most amount of ATP can be consumed over the longest period of time. Burning through too much glucose too quickly is the equivalent of burning through all of your wood so that you don't have any later when you run out and the temperature falls. Hopefully that makes sense. I know I'm going on a little bit more about this than I probably need to. Hopefully that makes sense. Okay. One of the primary things that regulate energy levels in a cell, one of the primary things that regulate the breakdown of glucose molecules are the energy levels themselves. So the amount of ATP and NADH, for example, are some of the key factors that actually regulate things like glycolysis and the Krebs cycle themselves. So what cells have actually evolved to do is use the presence, or I should say the relative levels of ATP and NADH, for example, as sort of an indicator of how much energy is actually in the cell at that point in time. So when ATP and NADH levels are high, that's an indicator that no more energy is needed at that particular point in time. It's kind of like your thermometer on the wall tells you that the temperature in your house is 75 degrees, and you don't really need to burn that extra piece of wood just yet. ATP is high, NADH is high, energy is sufficient for the moment. Conversely, when the levels of ADP and NADH levels are high, that's an indication that energy levels are low. ADP and NAD are the products of the breakdown of ATP and NADH. And high levels of that indicate that you actually need to start burning some energy to generate more ATP and more NADH, the equivalent of your thermometer falling. Temperature is getting low, time to burn some more wood. That's the idea here. Now, there's a wide variety of ways that this happens. This is something that's slightly different, sometimes dramatically different in different species. Different animals do this in a different way. So there's a lot of a large amount of variation here. We're going to kind of look at some of the most common ways that this happens that are kind of central to most organisms, especially in the eukaryotic domain. So NADH, ATP levels, these are key. Other ones can be things like citrate, one of the starting point, for example, the citric acid cycle. Pyruvate levels can do this, acetyl coenzyme A levels can do this, a variety of other enzymes can do this, and so a lot of different factors can take place here, but we're looking here at some of the most common ones. Okay, backing up here real quick to one of our earlier slides in glycolysis. One of the important steps in glycolysis is the conversion here of fructose 6-phosphate to fructose 1,6-bisphosphate. So this occurs right here in the glycolytic pathway. During the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, so ATP is consumed here. And this is one of the early ATP consuming steps, part of the energy investment stage of glycolysis. So one of the things that cells have evolved to do is to sort of regulate the expenditure of ATP at this point in order to only utilize that ATP if this fructose 1,6-bisphosphate is going to be needed in order to go through the further series of reactions here to produce more ATP. Now, the first reaction here using the first ATP sort of traps glucose in the cell, kind of keeps glucose levels intracellularly, can't say that word, keeps the intracellular levels of glucose high. There we go. And that in itself is sort of important. So one ATP is sort of expended early to kind of trap glucose in the cell, and then another ATP expended here sort of commits this molecule to the further stages of glycolysis. Okay, so this is kind of a key step. Wasn't one of the ones I listed earlier, but it's a key regulatory step that kind of commits this fructose molecule here to the further breakdown during glycolysis here. Okay. So back to where we were in the sequence here. This stage here, which is step 3 of glycolysis, represents kind of a key control point for glycolysis. And the enzyme here is phosphofructokinase, the enzyme that takes fructose 6-phosphate and converts it to fructose 1,6-bisphosphate. Now, this enzyme is referred to as an allosteric enzyme. Allosteric enzymes have a binding spot for both the substrate and one or more regulatory molecules, also referred to as allosteric molecules. The idea is their active site can be turned essentially on or off by the presence of these control molecules. And these control molecules can either upregulate, activate, or downregulate, inhibit, depending on the context. Okay, so for example, in this context, the enzyme phosphofructokinase is actually activated or upregulated by levels of ADP, adenosine diphosphate, and AMP, which is actually adenosine monophosphate. These are essentially the precursors to ATP. So if you refer back we talked about ADP. We didn't really talk about AMP, but these are essentially the precursors to ATP. When ATP is broken down and the energy is being utilized, levels of ADP and AMP are going to be higher. So what that basically means is that ATP is being spent and it's being used in the cell. So when that's happening, we're going to need more energy. So if we're burning lots of ATP, we're going to have these byproducts floating around, and that's an indicator to the cell that we're going to need some more energy. It's the equivalent of the temperature in your room going down when you're burning wood for fire. Okay, so the high levels of these indicate we need more energy. So the way it works in nature is that the presence of these molecules, a high level of these molecules, will actually bind to an alternative binding site on the enzyme phosphofructokinase, and that actually reconfigures the shape of the enzyme to actually get it to increase its activity level to convert more of the fructose 6-phosphate to more of the fructose 1,6-bisphosphate. Okay, so real quick, here's a better figure to demonstrate how an allosteric enzyme works. So in this figure, it's showing, here's an allosteric enzyme. With an allosteric enzyme, we have both an active site and an allosteric site. These are often referred to also as a regulatory site. So we have a site for the normal substrate and a site in addition to that for what's called an allosteric molecule. Now what will happen in the case of allosteric inhibition is the allosteric inhibitor, in this case the inhibitor as it's referred to, will bind to the site and actually change the configuration of the active site so that it now won't bind the substrate, or in some contexts it just won't bind the substrate as effectively. So it essentially either shuts the enzyme off or makes it less effective, essentially down-regulating its activity. Okay, so when this molecule is present, the enzyme ceases to function or function as efficiently. On the flip side, allosteric activation is essentially the opposite. In this case, the active site is not in the ideal configuration when the allosteric molecule is absent. But when the allosteric molecule is present, it then changes the active site to a more ideal configuration so the substrate will fit appropriately. So the idea is that the allosteric molecule will either deactivate, inhibit, or activate the enzyme in order to make it either function less effectively or more effectively. And that's the idea behind allosteric enzymes. Okay, so in this context then, ADP and or AMP can both act as an allosteric activator, meaning that their presence increases the rate of the enzyme. And what this figure here is trying to show is that a dark green arrow indicates a stronger, more active presence in the glycolytic pathway. Okay, because these levels indicate we need more energy, like a low temperature in the room. On the opposite side, high levels of citrate and ATP are indicators that we've already got quite a bit of energy here. If we've got a lot of citrate, that means we're converting pyruvate into acetyl coenzyme A, and acetyl coenzyme A is being generated into citrate during the Krebs cycle, citric acid cycle. ATP indicates we've got a lot of energy already. These are the equivalent of having a high temperature in the room. We don't really need to burn any more fuel here. So those indicate, these bind to the enzyme, change the configuration to make the active site less favorable for the enzymes. Okay, so these are not necessarily a black and white, they're going to bind or not bind. They tend to change it so that they're either more effective or less effective at binding the enzyme, and thus increase or decrease the rate of activity. Okay, so what that does then is it kind of sets the pace for the rest of the steps in glycolysis. Going back one more time here, we're looking at this step right here, and we can see that immediately after that we've got a whole other series of reactions that are going to take place. Okay, and each one of these other reactions is going to generate more energy ultimately. So by controlling this sort of step here, we're kind of going off the ledge, so to speak. If we can control the rate at which we go over that edge, we can kind of set the pace of all the other activities in the glycolytic pathway. So this tends to be a key regulatory step in glycolysis in virtually all eukaryotic organisms, and that's because it requires an input of energy, and all the other steps hereafter will follow if this step takes place. So by controlling that one step, we control a large portion of the glycolytic pathway itself. The key control point in the Krebs cycle, or the citric acid cycle, is actually the very first step itself, the step in which oxaloacetate along with acetyl coenzyme A are converted into citrate. So this step represents the really kind of the major initiation of the citric acid cycle. Obviously, it's the first step. And so cells have kind of evolved a control point right here at the very beginning. Okay, again, there's other control points in the Krebs cycle. This one tends to be one of the most dominant and one of the most central ones found in virtually all eukaryotic organisms. So with this one, we have a similar sort of activity, different enzyme, but we have allosteric enzyme here. Certain molecules will actually increase the rate of activity. Certain molecules will decrease the rate of activity. Okay. In this case, high levels of ATP, NADH, and acetyl coenzyme A all inhibit citrate synthase, and that's the enzyme here that carries out this conversion reaction. On the flip side, high levels of ADP, AMP, NAD without the H, the unreduced form, and then simply acetyl without the coenzyme A, so the carrier without the, the coenzyme A without the acetyl, just the carrier itself. High levels of all of these, the opposite of each of these essentially, will upregulate the enzyme. Okay, so it's similar to what we saw here, where we have high levels of one activate, high levels of the other deactivate. Same thing here. This one uses NADH in addition to ATP, as well as the coenzyme carrier, along with the acetyl coenzyme A. Okay, so I gave some specific examples there. There's lots of other examples. They all pretty much focus around a simple premise, though. And the idea, as I kind of already stated early, is if you have lots of energy currently, you're going to waste energy by generating more at that point in time. So like a thermostat, you want it to change in reference to the temperature in the short term. We don't want to burn more energy unless we have to at that particular moment. We want to save the energy so when we need it later, it's available to burn. And cells operate on the same premise. So the shift from energy can take place in reference to the products of the cell, the relative level of those products, the primary ones being ATP and NADH. But this can take place in a wide variety of ways, and those are just kind of the tip of the iceberg and how some of those pathways operate. But it gives you an idea of kind of how that takes place. Okay, last part here. As I mentioned, we're kind of wrapping up some loose ends. Slightly different topic, but another kind of different angle here, loose end to tie up, is what happens when you don't have oxygen. And what happens when you don't have oxygen in the context of respiration is referred to as a fermentation reaction. Okay. Now there are some alternatives. There's what's referred to as anaerobic respiration. If you're in my lecture class, I might have mentioned that. Anaerobic respiration is a little different than fermentation. But what they both have in common is that they both take place in the absence of oxygen. Fermentation is a reaction that essentially revolves only around glycolysis. And what happens is when we don't have oxygen, we're not able to fully metabolize glucose or any of the other intermediates fully into pyruvate into acetyl coenzyme A, and then into all the intermediates of the Krebs cycle, and ultimately convert that energy through the electron transport chain. So what happens is we're kind of stuck in glycolysis because this is really one of the only stages in the whole process that doesn't require oxygen. So you can essentially conduct any stage of glycolysis, any part of glycolysis, without oxygen. As soon as you get into the other stages, however, you're ultimately going to need oxygen, at least to make those effective. For example, the Krebs cycle, you don't necessarily need oxygen, but you'll waste energy if you don't have oxygen, because all the products of that ultimately are going to require oxygen later. So essentially, you'll need oxygen in order to be efficient once you get past glycolysis. Okay, so to summarize here, fermentation is a reaction that can take place, a series of reactions that can take place without oxygen, and can still produce a net positive yield of ATP per glucose. Okay, and what happens is fermentation will take place when organisms don't get enough oxygen, or when they don't have oxygen at all, and it's a way to essentially survive in an environment without oxygen. And this is referred to as fermentation. Now, this is different from anaerobic respiration. We're not going to get into that. So if you're looking for some more on anaerobic respiration, there are other resources out there, but this is a conversation about fermentation, which is really just a reaction centered around glycolysis in the absence of oxygen. Now, what happens here, organisms have evolved to use this process in order to get by without oxygen, as I mentioned. So some organisms rely exclusively on this. Lots of bacteria are well known to utilize fermentation as their primary source of energy. A variety of species live in some of the deep, muddy environments, several feet down in the soil, for example. You'll find microbes, 100 feet down in the soil, if you can dig that far down. Under normal conditions, when they're that far down, there's virtually no oxygen down there, and they can survive in these environments. And one of the ways they survive is on fermentation reactions. Okay, so these microbes make a living every day in an oxygen-deprived location where fermentation is their primary source of energy. Some organisms can live in an oxygen-rich environment, but still, for various reasons, primarily evolutionarily, prefer fermentation. This goes for yeast, for example. So yeast can actually thrive in both oxygen and anoxygenic environments. And then some organisms, like you and I as animals, we actually prefer aerobic respiration because it's more efficient, but under certain conditions, we actually can produce more energy when we are requiring excess oxygen. So for example, as an animal, during intense aerobic exercise, our muscle cells actually become starved for oxygen, something you're familiar with. If you're running, if you're wrestling, if you're working out, your muscles are burning more oxygen than what they can get through your lungs. You're breathing hard, you're working out. And what ends up happening is your cells will actually go into a fermentation reaction to get a little bit of extra energy to make up for what they're lacking through your aerobic intake. So you're not getting enough oxygen. Muscle cells primarily will go through fermentation to burn a little bit of extra energy in order to make up the difference for what they need. Okay? So fermentation can be the primary source, a preferential aspect, or used to kind of supplement in certain scenarios where you're not getting enough to start with. Okay, so this shows here kind of the starting glycolysis, and we see glucose is converted to pyruvic acid. If oxygen is available, it'll go through the Krebs cycle, electron transport chain, producing a net yield of 36 ATP in a eukaryote. We talked about 38. 36 is a common amount in eukaryotes. When no oxygen is available, however, that same pyruvic acid, pyruvate, instead of going into the other stages, can be converted here into anaerobic fermentation. Anaerobic fermentation then will produce things like lactic acid or other fermentation products, which we're going to talk about next here. So fermentation is partly a solution to a problem, the problem being you want ATP, but you don't have the oxygen to generate it via aerobic respiration. So the problem it solves is it generates ATP. You get 2 net ATP per glucose, a small amount, but yet sufficient if that's all you're capable of doing. So in other words, beggars can't be choosers. You'd like to have the full amount of ATP that you can get through aerobic respiration, but since you can't get that, you're going to take the 2 ATP per glucose that glycolysis will generate to you, and you can actually make a living off that if you're an anaerobic fermentative organism. Okay, so you get a little bit of energy. It solves the problem of not being able to generate energy at all in the absence of oxygen. One problem is that one of the problems that it creates, however, is that it doesn't immediately allow for the regeneration of NAD like you would with aerobic respiration. So in aerobic respiration, we generate NADH as one of our electron carriers, as we talked about. Will dump its electrons to the chain. That chain will activate ATP synthase. ATP synthase generates more ATP. Excuse me. But during the transfer of NADH, the electrons from NADH to the chain, during that transfer, we automatically regenerate NAD+. Let's back up here real quick to give you that visual. NADH transfers its hydrogens and its electrons to the first enzyme in the complex here. That automatically generates NAD+. That's our starting compound. That's what we're ultimately going to use to regenerate more NADH later on. That's what we see here, for example, in a reaction where we take NAD and use it to make NADH, as we see here. So when oxygen is around, we automatically convert NADH back into NAD, and these cycle back and forth to allow the process to continue. When there's no oxygen, the problem is that we end up with NADH but don't have an immediate way to convert it back into NAD, which is necessary in order for this step here in glycolysis to take place. So this is a step in which glyceraldehyde 3-phosphate is phosphorylated and NADH is generated in the process. Okay. Now, what happens in fermentation is rather than dumping the electrons and the hydrogen here from NADH to the electron transport chain, what instead is going to happen is the electrons and the protons here from NADH are instead going to be converted into another organic molecule. So rather than dumping those into the electron transport chain, they're going to be dumped into an organic molecule that's going to act as the recipient of these excess components here. So in other words, you got to get rid of the NADH. You got to get rid of this H here, excuse me, which consists of electrons and protons. You got to get rid of that to regenerate NAD+ here. Instead of doing it to the chain, you're going to do it to an organic molecule. And that's one of the major differences in fermentation. Electrons and protons are dumped to an organic carrier rather than the electron transport chain itself. That is going to free up NAD+ so it can be used again. So let's take a look at how that can take place. So as it says here, fermentation donates electrons from NADH to organic compounds. So for example, when we produce pyruvic acid or pyruvate in glycolysis, the pyruvate itself, pyruvic acid, can actually serve as a dumping ground for the excess electrons and protons from our NADH. So pyruvic acid can essentially receive these subatomic particles, electrons and protons, and be converted into a different product in the process. In other words, pyruvic acid is reduced, meaning it gains electrons. And in the process, NADH is oxidized back into NAD+. So now we have NAD+. It can come back up here to where it starts and be used to generate more NADH. That allows for the phosphorylation of glyceraldehyde 3-phosphate to generate more ATP. Okay, so it allows for the cycling of NAD and NADH. Now, pyruvic acid, in the case of fermentation, is essentially the end product. So we're not going to take pyruvic acid and then turn it into acetyl coenzyme A, and then take acetyl coenzyme A and run it through the Krebs cycle. Instead, pyruvic acid, pyruvate, is essentially the end of the road in our fermentation reaction. And what ends up happening in a fermentation reaction is when it receives the electrons from NADH, it becomes one of a variety of waste products, depending on the type of fermentation reaction that we're talking about. So different organisms conduct different types of fermentation reactions. They're all slightly different. And one of the differences is the type of organic compound that we generate when we dump these electrons to pyruvic acid or pyruvate. So two of the most common types of fermentation pathways are lactic acid fermentation and ethanol fermentation, or alcohol fermentation. So there are slightly different variations of enzymes used, and each one produces a different compound in the end, for example, lactic acid or ethanol. Ethanol fermentation is one that we're familiar with in that it's used in baking bread. It's also used in the production of alcohol, such as beer and wine, primarily. Ethanol fermentation is common in yeast, which is why we use yeast for baking and for fermentating wine and alcohol. What happens in this case is pyruvic acid, our final compound of glycolysis, is converted by an enzyme into a molecule called acetaldehyde, a 2-carbon compound. And in the process, CO2 is given off. So we take some pyruvate, convert it to acetaldehyde, carbon dioxide is given off in the process. Acetaldehyde is then converted into ethanol, which is actually our alcohol. So alcohol is actually an organic compound that contains this OH here, which is partly what gives it its properties. So ethanol, alcohol, is created through a fermentation, and a high concentration of this stuff right here is what gives alcohol its effect. Now, one of the other things that takes place is CO2 is given off. This can make, certain beers, for example, have a bit of a carbonated taste, carbonated texture to them. Part of that comes from the retention of the CO2. Wine typically doesn't retain the CO2. But the CO2 itself is a byproduct of the conversion of pyruvate to acetaldehyde. This also is what gives the bread its rising effect when you're baking with yeast. And if you're wondering, why don't you get drunk off of bread? It's because you're burning off all the alcohol in the oven. Alcohol burns off well below the baking temperature, so you're burning off the alcohol. There's not anything left in the bread when you're done. In the fermentation process, you're trapping that alcohol and concentrating it to a high extent. That's ethanol fermentation. Pyruvate is converted through a couple series into a 2-carbon ethanol. Ethanol, just to back up real quick, ethanol, I just want to point this out. Ethanol is a waste product to the cell. So to a yeast cell, ethanol is essentially like their poop or their urine. To give you a nice visual there. It's waste. So they have essentially taken all the energy they can get out of the glucose and scrapped what was left as this waste product here. Okay? And as they say, one person's waste is another person's treasure, and that's kind of how ethanol works for those of us who convert it into a wine or beer, or use it to make bread for something like that. The breakdown ultimately creates a waste that the cell no longer needs. And during fermentation, this waste has to go somewhere. So cells have another process of kind of excreting the waste, getting rid of the waste, and eliminating it. That's a whole other kind of process there. But ethanol represents a waste product for the cell. The same is true of lactic acid fermentation. In this case, rather than generating ethanol, it's a different fermentation pathway that also starts with pyruvic acid, but in this case, it's going to convert it into a lactic acid product instead of an ethanol product. Different enzymes take place here, different contexts. So, for example, lactic acid fermentation tends to take place in animal cells. Also, certain types of bacteria produce this. But this is what will take place in your muscle cells when you are exercising or under intense aerobic exercise. What happens here is our muscle cells have evolved mechanisms to utilize this pyruvate as an extra source of energy if we can't break it down with oxygen fast enough. So in the case of lactic acid fermentation, you're running, you're exercising, you can't get enough oxygen into your cells to burn all of that pyruvate fast enough. So you're essentially backed up. But your body wants that extra energy like right now. So rather than waiting for the oxygen that's not coming in fast enough, the body will take this pyruvate, which is building up in the cell, and skip all those extra steps, which take time, and just immediately convert it into lactic acid, and in the process, convert a little bit of energy from glycolysis. So we're going to burn some of that energy there and get rid of the waste product in the form of lactic acid. Okay, so we're kind of jumping that excess energy into use without all the extra steps that go along with it. And when it's being created is the lactic acid. Lactic acid is a byproduct once again. And one of the things that happens when we exercise is our muscles tend to get sore, and that has at least partly to do with the buildup of lactic acid products in our muscle tissue. So when this lactic acid fermentation takes place on a high level, we get a buildup of lactic acid that makes us sore, and we actually start to feel that. And one of the things that can happen as an athlete is you can actually develop tolerance over time and an increased ability to break this lactic acid down. And that's one of the things that helps you run longer, reduce muscle fatigue, sort of build up a tolerance to exercise, is how your muscle cells deal with lactic acid, eliminating it faster and breaking it down quicker. All kind of goes along with sort of building up strength and endurance when it comes to being an athlete. So that's lactic acid fermentation. Okay, that is the last part there of our cellular respiration topic. You've got 3 videos to go over. You may have to go back and take a look at some of these topics separately. As always, email me if you have any questions, or bring those questions to class and we'll go over them next time I see you. Have a great day.